Coming. My name is Jian Ma. I am currently an associate professor in the computational biology department within the School of Computer Science at Carnegie Mellon. Sorry for the very general title. So I will talk about some of our recent work in studying nuclear genome organization and also how to do that comparatively across different species. I think this topic will be a little bit different from the rest of the talks that you have heard so far, and then probably for the rest of the week. So hopefully this is interesting to you. So the human genome, we all know that 3 billion base pair long, right? And then previously Mike had a great talk on how to assemble these sequences, and then a lot of effort actually in the genomics community is to, once we have this genome, how do you annotate them? Where are the genes? And more recently, people are focused on understanding the non-coding regions and where are the regulatory sequences. And then if you change a base pair, what's the consequence of that? But this human genome, if you linearly you stitch all the chromosomes together, it's about 5-6 feet long on average. And then I think but it is actually within the cell nucleus, which is about 5 micrometer, the size of 5 micrometers. So it's all these chromosomes are packaged and folded into the cell nucleus. But we have very limited knowledge how these chromosomes are folded into the nucleus, and then what's the connection to function. And then also, in human genome, right? You think about we as humans compared to other mammals or non-human primates, what has changed in the course of evolution? So these are the questions that my group has been particularly interested in the recent couple of years. So genome organization, it has, if you look at in different resolutions, yesterday Frodem gave a great talk in studying the topological associative domains, a structure that can be observed by looking at these high C contact matrices. But at the highest resolution, you'll be able to these so-called chromatin loops, and there are several different types. In general, I think there are 2 types of loops that have obtained the most interest and attention. The first is these the interaction between distal enhancers and proximal promoters. So in this figure, you can see that this is an enhancer, which could be very far away from the target gene, and then the chromatin can be looped to contact to interact with the proximal promoter to drive the expression of the target gene. And this is one type, enhancer promoter interactions, and there are a lot of players involved. Another type of loop is more, I think it's a bit more general. Sometimes it doesn't involve any genes at these anchor regions. These are mediated by these architectural proteins. So one of the most important one is those loops that are mediated by CTCF proteins. So Frodem also mentioned this yesterday. And one interesting observation in recent literature is that the majority of these loops, if you look at these CTCF-mediated loops, the majority of these CTCF binding motifs at these loop anchor regions, the majority of them are in convergent orientation, in the sense that these 2 motifs are facing each other. There are also motifs that are in tandem. They're facing the same direction, but the majority of them are in convergent orientation. I will talk about this in a few minutes. But these are just loops. If you step back, think about if you're looking at the cell nucleus right now, and then you step back, right? So you see lower resolution, but you see larger context. So in addition to these loops, these loops are the chromatins for these loops, but if you at a little lower resolution, you will start to see these structures, that these topological associative domains. But if you keep stepping back, you will see that these chromosomes also have these chromosome territories and or these nuclear compartments. Some of them are more active, and some of them are more inactive, and these can be also can be inferred from high C data. However, there's also one very important aspect of the nuclear organization is that these chromosomes are also contacting and interacting with other so-called nuclear compartments or other constituents in the nucleus. Right? These are, say, the the nuclear lamina, the nuclear envelope, and then there are also other complex structures in the nucleus that are serving very, very important roles, say in gene regulation. For instance, there's a structure called nuclear speckle, and there's also nucleolus, centromere, and so on and so forth. So these kind of the chromosomes are folded into this three-dimensional space, and interestingly, intriguingly, this folding and packaging is highly regulated. So if something goes wrong, it may lead to disease. And Freudian also give some a nice example in the talk yesterday. But I want to show you one example that is kind of different. So it's about the nuclear structure itself. So this is the famous example of this nuclear lamina, and there's a mutation on this gene called lamin A. If there's this mutation, then the may result in unstable nuclear envelope that progressively damages the nucleus. So this is relevant. This is the genetic cause of this progeria disease, and it's also relevant to aging. And then progeria patients typically die as teenagers. As you can see, this is the normal cell nucleus, but this is the disease state. But certainly there are other structures in the nucleus that and then their roles are starting to people are starting to understand their roles in development and also disease. So here's an outline of my talk. I will give you a very high-level introduction of some of the projects we did in the past couple of years. Some of them are, I think I provided two related references, is this one and this one. When I provided links, they're still on bioarchive under review, but right now they have been published. And so first I'll talk about these loops, and then I'll spend probably more time on a method we developed recently to model these continuous functional genomic signals across different species. And we use DNA replication timing as a proxy to look at the genome structure across different species. And if I have time in the end, I'll talk a little bit about our ongoing work. This is unpublished, but hopefully the preprint will be submitted soon. So this is like appetizer. This will be the main course. There's a little bit of dessert if we have time. So first, so we are interested in building some kind of predictive model that if you have this, you'll be able to predict the actual enhancer-promoter loops. So the question is the problem setting is very straightforward. If you're given two regions in the genome, right, you may have some other information, say this histone mark, ChIP-seq data can tell you where the putative promoters and putative enhancers are, like H3K4. Monomethylation will be able to tell you where the promoters are. H3K27 acetylation will be able to tell you where the putative enhancers are. If you have these information, and can you build a model to tell people if this putative enhancer in that promoter is going to form a loop or not? And this problem has been studied before using using signals from functional genomic data. So this target finder is a method developed in Katie Polo's group in UCSF. The method uses many functional genomic data sets like DNA-seq, histomark ChIP-seq, and transgene factor ChIP-seq, and so on and so forth as the input features, so that you'll be able to build a predictive model. You train the model. You have the training set that measured from high-C interactions. So if there is a interaction, you say that there's a loop, there's a positive sample, and if there's no loop between the two enhancers, between these pairs, and it will be a negative sample. And there's a similar method from it's called a Repli from Soshimi Naroi's lab in University of Wisconsin. Also uses functional genomic data sets for features extraction. The purpose is very similar. So the main finding from these two prior work is that signals from functional genomic data are informative to computationally distinguish the ones that are a pair of enhancer promoters that are interacting and the ones that are not interacting. But we're interested in a slightly different question. So assuming that you forget about all these functional genomic signals that are involved in the surrounding regions of enhancer and promoters, if you only look at the sequence-based features, can you build a predictive model using only sequence-based features to do that kind of predictions? So the two questions: are there sequence determinants for promoter enhancer interactions? Are these sequence-based features sufficient to accurately predict these interactions? So in this work published in ISMB last year, and then so we developed this method called PEP, predicting enhancer promoter. There are two modules. One is very the methods. So all the methods I talk about today are very simple, I think. So, but I think the question itself is probably more interesting. The context itself is probably more interesting. And then I'll tell you how we adapt to say some existing techniques in a different context. So first, there are two modules in this PEP work. So first, the motif is very straightforward. You just calculate the motif occurrence, or we use to normalize the motif occurrence as features. And the second module is based on this technique in natural language processing called the word embedding. In particular, we use these word-to-vec method that's been widely used. So the goal is that you embed each word. Like in our case, we have a k-mer, right? So k is 6 here, each k-mer, and you embed that into a new n-dimensional feature space, and n can be selected or trained. And here, I think eventually we use n as 300. So each word, each k-mer in your sequence will be able to embed that into a 300 dimensional feature space. And word2vec in essence is a neural net, is a 2-layer neural network. You can train to predict the target word. Here is the target word, W is the target word, C is the context, meaning that you have a target k-mer and also the surrounding region, the k-mers that are surrounding this k-mer. You want to make sure that given the context, you'll be able to predict this target k-mer. So the objective function is you have this theta is the word embedding model you're trying to train, and you want to maximize this objective function, which this part is all the pairs of the target word and context pair, and this is to maximize this part and also minimize this part, which is the noise word and context pair. So so we have these two parts, and then eventually we actually integrate the features from these two modules. So the rationale is that for motifs, for the known motifs, we want to find out if the known motifs are involved in mediating these loops. But in addition to the known motifs, there might be some other sequence features that are not captured by the known TF motifs that can be captured by the word embedding model, by the word2vec. But if you put them all together, you expect that you may be able to achieve high performance. So this is what we did. We compared with the target finder. Using the same data sets, same evaluation approaches, and we look at different metrics and then across 6 different cell types. So the conclusion is that the performance is comparable to target finder. So recall that I mentioned that target finder uses many functional genomic data sets, but in our case we didn't use any of the functional genomic. We only used the sequence-based features. So it shows that sequence-based features alone can achieve comparable, but it looks like the functional genomic is still little bit more informative in most of the cell types, but at least the performance from the sequence-based feature classifier is comparable. And also the other trend is that is as expected, in general, the PEP word embedding module is doing a bit better than the motif module, PEP motif module, and then this integrated module is also doing better than individual PEP motif and PEP word modules. Yes? Could
you explain a bit more what's being evaluated? Like what's y-axis?
Sure. So these are different metrics. So again, the testing data. So for each cell type, there's high-C data has been generated. So for each pair, you will be able to see if they're going to form a loop, right? So if they form a loop, that's a positive sample. If there's no loop, then it's a negative sample.
Any loss you don't for a moment you don't know where promoter enhancer is. You checking it.
No. This is given. You know where they are, the putative enhancer and promoters. So that's also the input for the target finder. And then the goal is to distinguish these two types of interactions. But here I showed only in the training and testing, they're all in the same cell type. And that's the limitation of this approach. And then we tried this cross-cell type evaluation, but the performance was not that good. So we also looked at these CTCF-mediated chromatin loops. So as I mentioned, the majority of pairs are in convergent orientation. But for a pair of convergent motifs, that are bound by the CTF protein does not always form a chromatin loops. So the question is, if I give you a pair of convergent CTCF motifs, can you predict if they're going to form a loop or not? And so the questions we want to answer here is what are the main sequence level differences between the CTCF motifs that form a loop and those that do not form a loop? And second, for a certain cell type, can we train a model that predict those loops given a convergent pair of motifs? And third is, if you train a model in one cell type, are you able to predict a loop in a new cell type? So we want to do cross cell type predictions, because enhancer promoters, they might be very cell type specific, but here we try to see if there's anything that is shared between these different cell types. So a few years ago, we developed this approach. I won't get into details, but if you're interested, you can look at the paper. The goal is to try to give a timing of a cis-regulatory element in the human genome. So if you observe a chip-seq like between human and mouse, it looks like they're shared peaks, and then you may say, oh, this motif underneath this GATA1 chip-seq peak might be conserved in mouse, right? But if you look more closely, it may not be the case. It could be that both of these motifs, these events emerged after human-mouse common ancestor. So the model itself is trying to reconstruct the most likely ancestral state at each node in this tree in terms of number of motifs. So you want to you try to maximize the likelihood, right? Given the model itself and the probability of observing all these motif occurrences in the leaf nodes. So here, as you can see, that these highlight ones are the GATA1 motifs where they are in alignment, and the model tells us that there's actually two events that this motif emerged here before primate common ancestor, and also here before mouse and rat. And why this is useful? Because we actually thought about, maybe this is something that if you want to look at the motif, if it's look at its conservation across different species, we can actually use this score. We call it branch of origin score to quantify when, how old this motif is, right? So maybe the older ones are more likely to form loops, and it turns out that this is true. And then so we again, I won't introduce the method because this is very similar to the word-to-vec model I introduced in the first work. And as you can see that if you only use word-to-vec, you can achieve pretty good performance in terms of AUROC and AUPR. And if you only use the branch of origin, the information I showed you earlier, when this motif, older ones are more likely to form loops, and you can still get fairly good AUROC and AUPR. And we certainly tested other features, like other sequence features, for instance, CG content, GC content, and say the distance between the two pairs, although we control that it has to be within a certain range. And we also add these chip-seq of CTCF signals and the DNA-seq signals from the surrounding regions. And certainly if you add all of them, you can achieve very good performance. So here's one example. And this is in GM12A6A78. Here it is the real loops of the CTCF-mediated chromatin loops in this cell type. So these are the loops, and this is the true positive. So this is the predicted ones that are true, that are agreeing with the real answer, right? And we also have some false. You can see that we have a low number of false positive and false negatives. And you probably cannot see that more very clearly, but here there are two tracks that shows the branch of origin. One is more recent, after primate common ancestor, and more is one ancient, before primate common ancestor. We show that the ones that are that came into the human genome before primate common ancestor are more likely to form these chromatin loops. So the next question is, can we train a model in one cell type and then predict in the other, and what the performance would be? So it looks like we can actually do that. But one thing that we did this a bit more carefully, that we don't want to have the same training and testing. So all the the training and the testing in these two cell types, they're all cell type specific. In the sense you train a model using cell type specific loops in one cell type, and then you predict loops, try to predict the loops for the cell type specific loops in another cell type. And you can see from this plot that the cross cell type, there's some variations, but overall the AUROC is reasonably good. Show you one example. This is the model is trained in GM cell line, and then we are doing the prediction in HeLa cell. So these are the loops in GM cell line, and then these are the loops in HeLa cell. So these are the real loops in HeLa cell, and then highlight ones these are the cell type specific ones, right? As you can see that we can still predict these cell type specific ones accurately, but there are also some small number of false positives. So summary so far. So we developed these method to based on, for instance, word embedding to help us predict these chromatin loops. And you may ask why these models are useful, because actually word embedding, there's one limitation is some of the features are hard to interpret to what they are. But I think it's still very, potentially it's useful to help us predict the consequences or the importance of these non-coding variants. For instance, you can build a model in some normal cells, and then in cancer cells, you can see if the somatic non-coding mutations is going to disrupt the long-range interactions. So those kind of models will be informative. So if you think about these genomes, you know, if you compare human and other mammalian species, right? So a lot of interest has in the past decade and 2 decades also, you know, people are developing these methods to compare these sequences to identify point mutations, small indels, large-scale rearrangements, and also building models for studying cis-regulatory elements, cis-regulatory modules across different species. And in order to you know, link the sequence level changes to phenotypic variations. But you know, for instance, our recent work is to look at these large-scale rearrangements across our mammals, and we highlight the ones that are the number of breakpoints that you can observe on the branch from all the way from Ethereum common ancestor all the way to human. But where's the role of high-order genome organization, or at least can we develop some methods to help us interpret those data that can give us some insight how the genome organization has evolved in mammalian evolution or in, say, in primate evolution. And this is the recent work that we did in utilizing DNA replication timing data. So DNA replication timing, right? In interphase, in the S phase, the DNA will replicate, and this is the sort of the reflexive temporal order of the reflexive temporal order of the DNA that's being replicated. And earlier work has shown that early replication takes place in the interior of the nucleus, mostly. And replication at the periphery, and also there's a structure called nucleolar periphery, takes place in the second half of the S phase. So they can be considered as late replicating. And this general pattern is very much conserved across different species. And there's also early observation demonstrated that DNA replication timing reflects spatial position of the chromosome. So essentially, if the chromosome is more towards the interior of the cell nucleus, the DNA on those regions will be replicated early during this interface. And there are some observations early on between human and mouse in the mouse ENCODE project. So to look at these genome-wide patterns of conservation and dynamics in a pairwise comparison fashion. And actually, they found that, you know, DNA replication domains aligns very well to the TAT boundaries, and so those are one of the major observations from that work in this Pope et al. Nature 2014. But in general, you know, if you have multiple species, right, in mammals, especially in non-human primates, for instance, you want to compare human with the more recent relatives, and how can we do that? And then actually, there's no data set that's available. So we generated a DNA replication timing data set in this work in collaboration with David Goba's group in Florida State. So I introduced the data set a little bit first. So these are called Repli-se. So you can map the replication timing program in the genome-wide fashion. So in this kind of protocol, so you have these the cells are asynchronously cycled, and they're post-labeled with BRDU. So the BRDU can be incorporated into newly synthesized DNA of replication cells, and then you can do cell sorting to separate them into two fractions. One can be considered as late replication, and the other one is early replication. So so through sequencing, you'll be able to get these reads. So the first step of processing these reads is to map these reads to the genome. And for each fraction, you have reads from early fraction, and you have reads from late fraction, right, after you map to the reference genome. So certainly there's a window size, and then typically it's 50K or 60K here we used. And then we take the log2 ratio as the replication timing signals. So how to interpret these plots? You will see quite a few of these later on in the slides. So if it's greater than 0, then it's considered early. And if this part of the plot is considered late. So if the signal is higher, the higher the signal, the earlier it is. The lower the signal, the late replicating that particular region of the DNA is. So the question is, how can we, if you have these type of data from these multiple species, like in this case we have 5 different species: human, chimpanzee, orangutan, gibbon, and green monkey, and this is the phylogeny describing the evolutionary relationships among these 5 species, and you have signals from each one of these species, right? So you can already see that it looks like this first region, all of these 5 species are being replicated early, and the second, this region, all of them are being replicated late. But this one looks like human and chimp, they share early replication timing pattern, but other 3 species, they have late replication timing program. So can we, how to develop the model that actually consider these continuous features of the signals? And previously there's some work in the cross-species functional genomic data comparisons. And there's also models like these Phylo-HMM to consider the features in the genome-wide fashion across multiple species. But for Phylo-HMM, it doesn't really consider continuous signals. But for the models that consider continuous signals, it's not genome-wide. They're typically for single locus. So why we want to consider continuous signals? Because it gives you a much richer information or the more complete spectrum of how the signals are being distributed in the how to fully capture the signals. For instance, if you walk into a Starbucks, right, you can get a cup of coffee with these 4 cup size, right? Can you get a cup size with 13.68 ounce, right? So you can't, because these are the discrete characterization of the cup size for you, right? And then a lot of analysis so far, we found that for ChIP-seq, say across different species, people look at the peak gain, peak loss. So it's very discrete. Or you can say you have an intermediate state, right? It's kind of weak peak or strong peak, right? So, but these states are rather discrete. So our goal is that can we consider these signals as complete, continuous traits across these different species? And there's actually a lot of early work in this direction, and then we're building our method based upon these early work I will mention in a minute. So for instance, like here, our ultimate goal is to partition the genome into these different states, where each partition represents a different state, and these states represents a different evolutionary history of the signals across these 5 different species. So the model is actually quite simple, the setup. So in essence, still an HMM. So you have these X here is the same. So all right, so still HMM, and the states represent different phylogenetic models, and you have these transition probabilities between these states. And but the key is the emission here. So the emission here we utilize a Gaussian process to model the evolution of continuous functional genomic signals. And we use a model called OU model, Olsten-Uhlenbeck. I will introduce it on the next slide. And so for HMM, right, certainly we have these parameters, have these phylogenetic models, and then this A is the transition probability and pi is the initial distributions of the states. And then for this side, there's this phylogenetic model. There are a few parameters that we want to estimate, and they're all related to the OU process that we'll talk about in the next slide. So just give you a sense of what this model is doing. So these signals, right? These lines here are showing the hypothetical signals you're trying to measure. In our case, it's the replication timing signals, and a t is the time in the course of evolution, and a time t1 looks like there's not much change, but in t2 there's a speciation, right? So after there's a speciation, and looks like this black line is deviating from the rest. And after this t3, this branch, right? This signal is going way up than the rest of three. So this could represents the situation where, in human and chimp, right? In this one clade, the replication is early, but the other three, they're still late replication timing. And in the ancestral state, it's kind of late, but only two species are gaining changes in the signals. So this OU process can be considered as a modification of Brownian motion, which is a continuous time stochastic process with mean as 0 and variance as the increment of the time. And this actually has been used in several different studies, mostly in studying single locus phenotype evolution or gene expression evolution across different species. And the model itself is a combination of a deterministic part and a stochastic part. And the stochastic part is the Brownian motion part, and the deterministic part is this part that represents the tendency of a random walk to move back towards the optimal values. So this Brownian motion can be considered as a white noise here. It's the random variable. The derivative bt here is the it's independent and identically distributed normal random variables with mean as 0 and the variance is as the derivative of t. So for multiple species, we assume that the traits of the observed species follow a multivariate Gaussian distribution, and under the OU process, we can calculate the expectation covariance and the variance of the signals for each node in the tree. And this is really built upon this paper. I think this is from Rasmus Nelson's group a few years ago, looking at these gene expression evolution across different species. So now we can try to maximize the expectation of the complete data likelihood, right? You can write out, since you have these expectation variance and covariance, and then so some notations I won't explain in detail. Like this P is the immediate ancestor of the current node I, and A here is the most recent common ancestor of A and J. So we did the parameter estimation using EM, and then once you have estimated parameters, you can estimate the states in a genome-wide fashion using Viterbi. So I show you some examples, what the result looks like. So this is the predicted RT evolution patterns, replication timing evolution patterns. So in this region, if highlight regions, so these are the signals, right? These are the signals of replication timing. And we ended up with 30 states of the replication timing. We can certainly have more states, but we feel like 30 is a good balance. Like here, we separate them into conserved early, like across all these species, they're all early, and these are conserved late, right? We call them into these, even further group of them into for the convenience of analysis into 5 distinct groups. And sometimes you can't tell what the evolution pattern is. So, we call them non-conserved because there's some variation across different species. And if 4 out of 5 species, the replication timing is early, and the 5th one is kind of in the middle, we call them weakly conserved. And similarly for weakly conserved early or weakly conserved late. So here's the distribution of in these 30 states, how the signals are distributed. So here, these 4 are conserved early, and these next 4 are conserved late. As you can see, there's still some differences in terms of their signal strength in some of the species. And here's the transition probabilities, and it's as expected. For those states that are close to each other, they're kind of all conserved early, and for the ones that are all conserved late, they have higher transition probabilities. And then we did a fair amount of a lot of analysis by comparing these different states to other annotations. Say, because this is actually, sorry, forgot to mention this. This is from lymphoblastoid cells, so the GM12878. So there are a lot of data from ENCODE, so in human. So you look at, these are the histomark ChIP-seq data, and also CTCF ChIP-seq. And these are A1, A2, B1, B2, B3. These are so-called Hi-C subcompartments. So these are the genome-wide clustering of the Hi-C signals, right? So you can separate them into these groups, and each group actually has some meanings. For instance, for A1, A2, they are generally considered as active regions, but for the B compartments, they are generally considered as repressed, inactive regions. I'll just give you one example. So, for instance, for these 4, they are all considered as conserved early, but we separate them. Like you may ask why you separate them into these 4 different distinct groups. But we thought that if you look at these by comparing with other annotations, you start to see that these separations may have something pretty interesting, even though at the moment we don't really know how to interpret them. For instance, this S4, this is also conserved early, but it's highly enriched in A2 Hi-C subcompartments. But for the other 3 states, they are mostly enriched in A1 subcompartment. Next, we compared. How much time? How much more time I have? How much? So next, we looked at the from this paper from David Gilbert's group to look at these replication timing program in ES cell differentiation. So you can separate these regions in terms of if they're all replicated early in all different cell types during differentiation, or they're all late. They're so-called constitutive early or constitutive late, and also the ones that are more developmental, meaning that they vary a lot. And we found that for the constitutive early replicating regions, they're mostly conserved early across primate species. Similarly, for conserved late, most of them are, majority of them are conserved late across different primates. And for the developmentally regulated ones, they're mostly non-conserved across different species. And we specifically look at these different states, lineage specific, and look at the genes that are enriched in these different states, and found that for difference, like for instance, for this state 9, is the human chimpanzee specific early replication timing regions, and they seems to have distinct functions of the genes compared to some of the other states. And similarly, for other lineage-specific states, you see some different enriched gene functions. So next, we ask, you have these patterns, and you see their differences across different species. And so, what is the sequence level contributions, right? So we don't know. So we looked at some of the known sequence features like transposable element sequences or TF motifs, and see if there's any enrichment. And there's earlier work showing that if it's ALU is more enriched in early replication regions, and this is also, we also observed that. But one thing that we don't know how to explain so far is these RNA repeats. So these are SRP RNA or these SN small nuclear RNA. They're highly enriched in conserved early replicating regions. So if some of you know the biology or have studied these RNA repeats, I think it will be interesting to talk. And we'll also look at some of the motif enrichment for TFs in different states to lineage-specific states. So the idea is that if you see a human-specific early replication region, we ask, is there specific TF motifs, so this element might be contributing to that. And we did see some signals, but it's still rather preliminary. Like for instance, for these human-specific early to the state 9, you see that there's a lot of ETS and Fox family TFs. Looks like their motifs are enriched here. So looks like I won't have time to go through the last part. And for the ones I talk about today, here's the list of the programs I talk about, and here are the papers. I didn't have time to mention this work. It's a new technique that in collaboration with Andy Bellmont's group at the University of Illinois to look at the nuclear genome compartmentalization. Essentially, is to look at the chromosomes and then their interaction with nucleolamina, nucleospeckle in the nucleus. There are some very interesting biologists we learned from these analyses. And so here are 3 students that contribute to the work I mentioned today: Yang Yuchuan Wang, and Ruochi Zhang. So they worked on the three projects that I covered in this talk. And also the collaborators, and especially Chuan Gu used to be at the University of Virginia, but now he's actually here in UCLA. Just moved to the computer science department here last week, and also the funding sources. Thank you very much.
So we have time for a few questions.
So you spoke about 2 processes, one deterministic and one would you mind go back to that slide with the Brownian motion?
Sure.
And I was wondering if in that slide,
This one?
no, the tree was the time, where you had the time.
This one?
Exactly. So the speciation events are the deterministic signal that you take from somewhere, some external information, and the Brownian motion is...
No, the optimal value, the theta, like on the next slide, it's actually also trained from the estimated from from the data itself. Yes.
Okay, so the speciation is also hypothesized.
Yes, it's estimated from the data.
So this should somehow estimate the brief phylogeny over these guys, over these species. The tree that you see here.
The tree, we didn't estimate the tree itself. We assumed the 2 topology is known, because otherwise it's too much.
Okay.
Yeah, too many possibilities. So we assume that the species tree is known, and what we estimate is the values, like the optimal values, the selection strengths associated with these different nodes in the tree.
And should there be any correlation with the species tree and the tree that you did here?
No, the tree is given. The tree is known. We assume the tree is known. We want to estimate the OU process on these different branches, and the parameters that are associated with these different OU process on each one of these branches in the tree. That's maybe we can talk about that.
Okay, sure.
I think we are out of time.